Okay. Okay, here we go. Very quickly. Very quickly. I just want to talk about everything that we have going on here. We have in the thymus. Okay, we're going to go through some things about the T cells, what's happening in the thymus, what's happening in the lymph node, and what's happening at the effector at the site of infection. Okay, so within the thymus, okay, this is where T cell development happens. Okay, so the hematopoietic stem cell is going through the bone marrow, and then a very early progenitor will leave, and then get attracted into the thymus by certain thymic cytokines and certain generalized thymic factors, right? Like thymopoietin and all that stuff. But anyways, we're coming to the thymus. We have a progenitor T cell. Okay, at this stage, this is a double negative thymocyte. We call it a double negative thymocyte. Okay, and we move on. We have a next stage. We have a pre-T cell. Okay, the pre-T cell. Okay, it has to in this stage, it has to express a a beta T cell receptor chain. Okay, a beta chain of the T cell receptor. Okay, if it cannot express a beta chain of the T cell receptor, it'll go through apoptosis and die. Okay, because these guys, they're programmed for apoptosis unless they get a survival signal. Okay, and interleukin 7 is really that survival signal that they receive. Okay, so it promotes survival. Um, okay, and now, so the pre-T cell, what did I tell you? It has to make a B TCR chain. How does it make that BTCR chain? For that VDJ recombination, okay? So VDJ recombination has to occur here, and that's from the VDJ recombinases, so the RAG1 and RAG2, okay? VDJ recombinases. Now, something else also, you have to have a surrogate protein here, and that surrogate protein is really there to test the signaling to see if this beta TCR chain that you made is even functionable, okay? So if it is functionable and it works with a surrogate surrogate alpha chain, then you'll send a signal down to the CD3 and the zeta chains, and then you're good. If it's unfunctionable, if it doesn't function, it's useless, you'll also go through apoptosis and die, okay? Next stage will become a double positive thymocyte. What is a double positive thymocyte? It has a CD8 positive and a CD4 positive on it. At the same time, now it has a fully functioning TCR alpha and beta. Okay, so it has an alpha chain and a beta chain. Now, from the pre-T cell to the double positive stage, you had to express a TCR alpha chain. Okay, a successfully made TCR alpha chain that works. If you could not do that, you would go through apoptosis and die. Okay. All right, so now we're at the stage here at a double positive thymocyte. Double positive thymocyte, okay, it can go through two things, positive and negative selection. We'll talk about what's happening here. Positive selection, okay? Let's go through a couple examples here. Let's say I have a CD4 positive cell right now, okay? CD4 positive, they're both double positive. But by random chance, the CD8 positive happens to just perfectly match with this MHC on this thymic stromal cell. If it's a CD8 positive, it's MHC1 molecule, okay? Let's come over here. Again, we have a double positive thymocyte. It has both CD8 and CD4 on it, but by random chance, the CD4 this time matches with MHC2, presented on the thymic stromal cell. Now, if this happens, if we randomly match, okay, match the CD8 with the MHC1 and randomly match the CD4 with the MHC2, what's going to happen is we're going to start down-regulating the CD4 on this one, and we're going to start up-regulating CD4 on this one, okay? And again, similarly, down-regulating CD8, okay? Just inhibiting that gene in general. And what's going to happen? We're going to have a mature CD8 positive cell, naive, of course, and a mature CD4 positive cell, naive. Very important piece that I want you to take away. This interaction, the T cell receptor and the MHC molecule, this has to be a weak interaction with the self-peptide. It has to be a weak interaction. If I come over here real quick, and I'll show you negative selection. You see that, again, this can happen in both MHC1, MHC2, CD4, or CD8. I'm just showing this in the CD4 stance right now. You see, if we have a very strong interaction with our own self-antigen, that's going to lead to apoptosis. Why? Because this could lead to a possible autoreactive T cell, and we don't want this, okay? So we get rid of it immediately, okay? Another thing that can happen with positive selection is if you have a failure to recognize an MHC and the peptide, then you will also go through apoptosis. So take a look at this T cell here, okay? Again, it's a double positive T cell, okay? Double positive T cell, and it has a T cell receptor, but take a look at this. The T cell receptor doesn't match the epitope on this antigen. And if you take a look here, the CD4 and the CD8s, none of them are actually recognizing this MHC molecule, right? So if you have a failure to recognize the MHC and the peptide, boom, apoptosis, okay? So now we know how we make our sort of thymic cells, okay? Now we have these naive mature CD4s and mature CD8s, okay? These guys can now go and leave the thymus. They can go into the circulation, okay? And they constantly do what? They constantly circulate in the blood and the secondary lymphoid tissue, trying to find an antigen, okay? Not trying to find an antigen, sorry. They're trying to find, well, they're waiting for antigen-presenting cells to present to them, okay? That's a better wording. So now let's go to the lymph node, which is a secondary lymphoid tissue. Within the lymph node, we have something very cool happening. In the lymph node, you see this dendritic cell? This dendritic cell is going to be, let's say now it has an antigen, okay? And it is presenting to this naive T cell, okay? What is that? That's a naive CD8 positive T cell, okay? So this guy, it came all the way from here. Naive CD8 positive T cell. Do you guys see this TCR complex? This blue thing is the T cell receptor. This is the MHC1, okay? And then that red thing is the antigen in between them, okay? Now, this first interaction is going to cause what we call signal 1, okay? Another thing is going to happen. We have CD28 on the naive T cell and B7, which is a co-stimulatory protein, on the dendritic cell. That B7 is going to interact with CD28 and cause what we know as signal 2. You might be thinking, okay, does B7, is it always just on the surface of the dendritic cell or an antigen-presenting cell? The answer to that is no, okay? B7, okay, B7, this co-stimulatory molecule that is on this dendritic cell right now. B7 is only expressed after an infection, okay? After an infection, which is why adjuvants are so important, because if we have a T cell, okay, and a T cell is going to get activated, it needs both signals. It needs a signal 1 from the TCR MHC complex, and it needs a signal 2 from the B7 CD8 interaction. If it only receives a signal 1 and no signal 2, okay, it will become anergic, okay? And if it becomes anergic, what does that mean? It'll eventually die, okay? There will be no T cell response and it'll eventually die, okay? That's why we need both signals, signal 1 and signal 2. So again, B7 is only expressed after an infection, right? So through that toll-like receptor pathway, which would release NF-kappa B. NF-kappa B would go and make this B7, and it would probably make interleukin 1, okay? So all this stuff, it's important. Okay, now B7 interaction with CD28 causes signal 2. You know what? You want to know something cool that happens? Okay, something really cool that happens is that after we activate this naive T cell, okay, newly activated naive T cell, one of the first things it does is that it'll express a fully made interleukin 2 receptor. Because normally the naive T cell has an interleukin 2 alpha, beta gamma receptor, but as soon as it gets activated by this signal 1 and signal 2, it'll make interleukin 2 alpha beta gamma. So it'll start making an alpha chain for the interleukin 2 beta gamma receptor, and it'll make an alpha beta gamma receptor. Okay? Now, at the same time it's doing that, it is secreting an interleukin 2. Interleukin 2 is an autocrine, okay? It has an autocrine action, and it'll act on the interleukin 2 receptor on the activated T cell, and now that T cell can go through expansion and differentiation. So it can proliferate and differentiate. Proliferate and differentiate into what? So it can proliferate, it can divide, but it can also differentiate into either being a cytotoxic T lymphocyte or a T helper cell. Okay? Now, if it's a CTL, then that's it. Okay, it'll go into the site of infection and it'll start killing, or find and start killing. If it's a T helper cell, a little bit more complicated. We need to go through further differentiation, and I'll get to exactly how that happens alongside here. But before we do that, let's talk about T cell signaling real quick. Okay, very quickly. So the T cell signaling, a couple of things are happening. T cell signal 1. Let's go through signal 1. Now, remember, over here we had MHC1, okay, presenting, it could be MHC1, MHC2, whatever. It's presenting to this T cell receptor, okay? And then CD8 positive is interacting with that. Something I want you to know, as soon as this MHC molecule interacts with the T cell receptor, okay, and it's for that specific antigen, and they fit, it's a perfect match, we're going to have what's known as the TCR CD3 clustering, okay? You know the CD3 and the zeta chains, those are your signaling proteins? The CD3s and the TCR complex, they're going to start clustering into one area of the cell. Once they do that, the CD4 positive or the CD8 positive molecule on the intracellular side, they're going to start bringing this kinase, okay, this phosphorylating kinase, LCK, close to this brown and this green thing. What is this brown thing? What is this green thing? This green thing is our zeta chains. This brown thing is our CD3, okay? So LCK, it gets brought closer to these guys. Once it gets brought closer to these guys, what is it going to do? It's going to phosphorylate the zeta chains, okay? And it's going to phosphorylate the CD3 chain. Once it does that, something else is going to happen. ZAP70, okay, this is a molecule, ZAP70, this protein, now it can come in and again, this is the phosphorylated ZD3 ITAMs, okay? ITAM stands for immunoreceptor tyrosine activated motifs, okay? So tyrosine-based activation motifs. And it'll, again, LCK phosphorylates the ITAMs on the CD3 and the zeta chains. Specifically though, the ITAMs that are phosphorylated on the zeta chain serve as a docking site for ZAP70. ZAP70 is going to come over there and bind. And now it's in closer proximity to LCK. LCK is going to come here and phosphorylate ZAP70. Once ZAP70 gets phosphorylated by LCK, it becomes activated. Now, this guy is now activated. What is it going to do? It's going to get us to make 3 very important transcriptional factors through 4 different pathways. Okay, so let's go through that. ZAP70 is going to do what? It's going to come here and phosphorylate all these guys, okay? Main thing you have to know, it's going to affect the PI3 kinase, and the PI3 kinase will lead to the PIP3. PIP3 gets activated and activates ACT and mTOR, okay? So ACT and mTOR, which incidentally increase protein synthesis. ACT and mTOR are going to give us AP1 transcriptional factor and NF-kappa B, but more AP1 specifically. The MAP kinase. The MAP kinases will also get activated by this activated ZAP70. And what that's going to happen is MAP kinases are also going to make NF-kappa B and AP1, okay? Another thing I want you to know, this one's very important, PLC gamma 1. What is PLC? Phospholipase, okay? PLC gamma 1 is going to do what? It's two pathways. First pathway is going to increase cytosolic calcium concentration, which will stimulate calcineurin. Calcineurin will start, will go down and it'll activate NFAT. What is NFAT? It's a transcriptional factor that we want, and it stands for nuclear factor of activated T cells. NFAT is primarily responsible for making interleukin-2. Okay, that's a transcriptional factor we want. The second pathway that you get from the PLC gamma 1 is going to be the DAG pathway. So diacylglycerol. Okay, and what is that? Diacylglycerol is just a lipid, right? So diacylglycerol, okay, it will get stimulated, and then that would activate protein kinase C theta. Protein kinase C theta will make NF-kappa B and AP1. specifically more NF-kappa B than AP1, but it'll still make a little bit of AP1. All of that we discussed, that was all signal 1. That was all signal 1, okay? All that was signal 1. Okay, very quickly, let's move on here. You know how I said we make a little bit of an interleukin 2 here from signal 1? And this is the reason why we need signal 2, is because interleukin 2 production increases by 100-fold after signal 2, okay? And what was signal 2? Signal 2 was CD28 plus B7. We showed it right here, okay? And B7 was on an antigen-presenting cell, and then CD28 was on the T cell. Another thing I want you to know is that B7, specifically, there's a reason why dendritic cells always present to naive T cells, is because they have a higher concentration of B7 molecules expressed on their cell surface than other antigen-presenting molecules, okay? Like macrophages or B cells, okay? Very important to know that. All right, that's that. One more thing about this signaling mechanism is that signal 1 transcriptional factors, like I said, they make interleukin 2 alpha chain, and that's enhanced by signal 2, okay? So signal 2 really enhances everything that signal 1 does, okay? And specifically 100-fold interleukin 2 production. That's that. Okay, that's what you need to know. Okay, so we talked about the T cell development, we talked about T cell signaling, and we talked about how it's getting sort of activated within the lymph node. Now let's talk about what I told you about before, is that the differentiation into T helper needs a little bit of more help. We need to become different types of T helper cells. And there's typically 5 different types that we're going to talk about here. T helper 1, T helper 2, T helper 17, and T follicular helper, and T regulatory, which we're not going to discuss in this video. We'll discuss in different lectures, but T follicular helper, we'll discuss its effector functions, but there'll be a whole dedicated lecture on how T follicular helper cells even get activated and what they do, okay? For now, we're just going to focus on TH1, TH2, and TH17, okay? Specifically, their activation. Okay, so let's get through it. There's 3 methods here that I want you to know, and you have to know this, okay? The first method is, in order for us to activate a T helper, it's really dependent on the type of pathogen we're dealing with, okay? So T helper 1 is really responsible for those intracellular bacteria. T helper 2 is really responsible for extracellular bacteria. T helper 2 is responsible for helminths. T helper 17 is responsible for extracellular bacteria and fungi, okay? It's written all here, guys. Now, another thing I want you to know is the fact that for the first scenario, intracellular bacteria, 2 things. The intracellular bacteria will be stimulating this dendritic cell, right? So dendritic cell will uptake it, and the antigen that it will present is a very specific antigen towards the intracellular bacteria. And then the intracellular bacteria will also stimulate NK cells. 2 things. They're going to release interleukin 12 from the dendritic cell and interferon gamma from the NK cell. Once they do that, what's happening? What's happening? Again, what is this? Right here, what is this red cell? This was an activated and proliferated T helper cell, right? So this is after interleukin 2 was secreted, guys. Okay, this is after this phase. Okay, this cell here, interleukin 12 and interferon gamma are going to act on specific gene transcriptional factors. In this case, it's going to be T-bet, written right here, T-bet. And that T-bet is going to initiate the differentiation of this T helper into a T helper 1 cell. Okay, let's move on. What if it was a helminth? Okay, if it was a helminth, we want to make a T helper 2 cell. How do we do that? You're going to have mast cells, you're going to have eosinophils, and you're going to have naive CD4 positive cells secrete interleukin 4. Interleukin 4 will act on this activated T helper cell, and it'll activate GATA3, the transcriptional factor GATA3. GATA3 will, of course, facilitate the formation of this T helper 2 cell. Let's move on to the third one. If it's an extracellular bacteria and fungi, we're looking for a T helper 17. A T helper 17, okay? But specifically, who's releasing these cytokines? Dendritic cells. Okay, so dendritic cells, they'll see an extracellular bacteria and fungi, they'll take it up, okay, and they'll present it, okay. Well, when they're doing this, afterwards, they're secreting a bunch of cytokines. What cytokines? Interleukin 1, interleukin 6, interleukin 23, tissue growth factor beta, okay? And these are all going to act on this activated T helper cell, and it'll activate RAR gamma T. RAR gamma T is the transcriptional factor that's going to facilitate the formation of T helper 17, okay? Boom, roasted. We've talked about the lymph node. We've talked about the thymus. We've talked about the T cell signaling. Now let's finally take a look at this effector functions at the site of infection, okay? So remember, guys, what's happened? Where's the journey been so far, okay? The journey was from the bone marrow, these guys came into the thymus, okay? And then they developed fully, and then they left through the blood, okay? And then they came and they would, circulate between the secondary lymphoid tissue and then the blood, okay? Let's say they go to the lymph node, which is a secondary lymphoid tissue. Over there, they get activated, okay? Once they get activated, they start proliferating and differentiating. Once they do that, then they can leave through the blood, and then they go to what? Where do they go? They go here. Effector function at the site of infection, okay? So they go to the site of infection. All right, so let's take a look at all these different sort of functions of these T helpers, and then we'll talk a little bit about cytotoxic T lymphocytes, okay? Okay. Effector functions at the site of infection. T helper 17, okay? So T helper 17, what did I say? It was for extracellular bacteria and fungi. T helper 17 can release two cytokines. Sorry. It can release interleukin 17 and it can release interleukin 22. Interleukin 17 is going to act on neutrophils, okay? So it's going to activate neutrophils even further, and it's going to recruit more neutrophils, right? So more mobilization. And that's going to help to increase our inflammation more than what we did in innate immunity, okay? Interleukin 22, when released from the Th17, it can go and act on epithelial cells, okay? And what it can do is, it can increase these antimicrobial peptides that we released before, defensins, if you remember, and it can also increase the barrier integrity, okay? So more tight junctions, let's say, within the gut, if we're talking about it, okay? And fun fact, really, if you want to know, people who have inflammatory bowel syndrome, it's really being caused by an excess of Th17 and the excess of interleukin 22 production, okay? Now, what about T helper 2 cells? T helper 2 cells, I said those guys were for helminths, right? So they were for helminths. Now, if they were for helminths, what do you think they're going to do? A couple of things, actually. Interleukin 5 is going to act on eosinophils, right? So it's going to activate the eosinophils, but interleukin 5 is also going to activate the Fc region, right? So the Fc receptors of mast cells and eosinophils. So it's not activating the mast cell. It's only activating the eosinophil, but it's activating the Fc receptor of the mast cell and the eosinophil, okay? So then they can lead to degranulation. Another thing, interleukin 4 and 13, it releases that, right? So it releases three cytokines, 5, 4, 13. Interleukin 5 is going to do that. Interleukin 4 and 13 are going to act on macrophages, and it's going to turn them into M2. What was that called when we turned them into M2? It was called alternative macrophage activation, okay? When a macrophage turns into an M2 macrophage, it'll start secreting its own cytokines, like interleukin 10, like tissue growth factor beta. And what is that going to do? That's going to initiate tissue repair, okay? And what? It's going to decrease inflammation. Another thing that interleukin 4 and 13 can do is they can go down into the intestines and they can increase mucus production and peristalsis, okay? And there's a reason for that, because you can have these helminths in there, and if you increase peristalsis, you're actually sort of squeezing them out, okay? All right, so let's move on to T helper 1. T helper 1 was for what? An intracellular bacteria, right? Intracellular bacteria and that's it. Intracellular bacteria. I'm just tricking you guys. M1, okay? It can make M1. So if Th2 could make M2, Th1 can make M1, okay? Very simple, guys, right? And how does it make M1? It makes interferon gamma. What did Th2 make? Th2 made interleukin 4 and interleukin 13 to turn a macrophage into a M2, which was called alternative macrophage activation. M1 is called the classical macrophage activation, okay? And it's done by interferon gamma released by Th1. When we have an M1, it's going to release its own cytokines again. It can make interleukin 1, it can make interleukin 12, it can make interleukin 23, and all of these are going to act to increase inflammation. Okay? Done. Finally, we'll talk about T follicular helper cells. The main job of these guys, I want you to know, is that they activate B cells. Okay? And I know I said effector functions at the site of infection. These guys, all of these guys, so the cytotoxic T lymphocyte, the Th17, Th1, Th2, they all go to the site of infection. The only one that doesn't is the T follicular helper. Those guys will go into the B cell zone, okay, or the follicles, and they'll stay there with the B cells, and they'll help activate them, okay? What cytokines does T follicular helper release? Interferon gamma, interleukin 21, and interleukin 4. We don't know what interferon gamma does. Interleukin 21, however, is for B cell activation. Okay, so it'll activate the B cell. Because you know how B cells, they can, they'll see their antigen and they'll be partially activated, but they can't proliferate and differentiate into plasma cells, right? They need a signal. That's from interleukin 21 from T follicular helper cells. Okay, so it'll activate them. Another thing that it can do is interleukin 4. It can switch it. Okay, it can switch the immunoglobulin that it's making into IgE, right? So B cell switch to IgE. And it's important that's very important when we're talking about a Helminth sort of situation where if you have a Helminth, your eosinophils and your mast cells, they cannot see that Helminth that's there, okay? They can't see it. They need it to be coated by antibodies. And once it is coated by antibodies, T helper 2 cell will activate those guys, right? What does it release? Interleukin 5, okay? It releases interleukin 5 and it activates those guys that they're FC receptors. And so those FC receptors bind to the FC region of the antibodies, okay? That was made by the plasma cells. How was it made? TFH secreted interleukin 4, which classed them into IgE, okay? And IgE, again, remember IgE for eosinophils, right? Are very specific and they bind to parasites, okay? So remember that, it's very important. Okay, so that covers these guys. And then very quickly, we'll talk about the cytotoxic T lymphocytes. All I want you to know with these guys, is that they can do direct killing of intracellular pathogens, specifically viruses, okay? So, and they can also kill cancer cells, okay? Just know that. Know that dendritic cells can come in there and present suspicious antigens that are from cancer cells, okay? Now, not important though. We have a CD8 positive cytotoxic T lymphocyte here, and you can clearly see it has a T cell receptor, and it has an MHC1 here, okay? And then what? That's the antigen over there, okay? And what's that thing binding to it? That's a CD8, right? CD8 positive is also binding to the MHC. It needs to recognize it. Okay, now, what are these guys, this yellow and blue thing? That's your ICAM1 and your LFA1. Those are your adhesion molecules, okay? Because you want to be able to hold it in place together while it's checking to see if that is a foreign antigen or not. If it deems that this is a foreign antigen, what is this guy going to do? It's going to start secreting two things, perforins and granzymes. Perforins, okay, what they're going to do is they're going to start making holes within the cell membrane of the cell, okay? And once they do that, the perforins actually facilitate the movement of the granzymes into the cell. Okay, so those pores that they made, the granzymes are going to move through them. What do the granzymes do? The granzymes are going to cleave and activate certain caspases that can lead to, That can lead to apoptosis, okay? So they basically activate pro-apoptotic genes, okay? And that's what the granzymes do. And they go into the cell and they do this. And that's going to cause cell death. There's also death receptors on each cell, and cytotoxic T lymphocytes have the ligand for it. If you really want to know, these green guys, these are just your generalized tissue cells, for example, and they would have the FOS receptor, okay, and then the T cell receptor would have a FOS ligand, so FOS-L, and the FOS-L would bind to the FOS receptor, and they'll also cause the intrinsic apoptotic mechanism without having to secrete perforins or granzymes. Okay, it's not a molecular biology course, so you don't have to know the exact mechanism. One more thing I want you to know is that this exact perforin and granzyme mechanism is the same thing that the NK cells use. Except the NK cells, they don't have MHC molecule, which means they're not really restricted like that. So let's say an intracellular virus decides to sort of downregulate our, downregulate our antigen on this MHC class 1. Is my T cell going to kill that? The T cell, is it going to see any antigen? No, it's not going to see any antigen, right? And it's like, oh, okay, there's no antigen, so this cell must be calm. This cell must be fine. And then it'll just leave, okay? But an NK cell is going to say, I don't see an antigen here on this, on this MHC molecule. I can't recognize it because the NK cell, it has an activator and an inhibitory receptor, right? And the activating receptor is always bound, and the inhibitory receptor is checking the MHC. If it senses that it's a foreign peptide that it doesn't know, or it's anything suspicious, so it's being downregulated, it'll immediately kill it, okay? And it'll use the same killing mechanism. NK cells do not give a hell, okay? They will do whatever it takes. Okay, guys, that pretty much covers everything. We've talked about T cell development, what's happening in the lymph node, T cell signaling, and then effector functions of the site of infection, okay?